everyone. Today we will discuss about the antibody structure and function. So let's begin with the class. So what is the antibodies? Okay, so antibodies is also known as the immunoglobulin, or you can say like it is the glycoproteins. Okay, so it is made up of carbohydrate and protein, so that's why it is called as the glycoproteins. Okay, and these antibodies are large y-shaped blood proteins which is produced by the blood plasma cells. Okay, and they bind to the foreign particle to invade them, okay, or to remove them. This antibody molecule have a common structure of four peptide chains. Okay, it is made up of four chains, which is it consists two identical light chain and two identical heavy chains. Okay, and each light chain is bound to a heavy chain by the disulfide bond. Okay, and other non-covalent interactions such as the salt bridges, hydrogen bond and hydrophobic interactions to form a heterodimer. Okay, so this structure of means glycoprotein, this is the antibodies structure is like that. Okay, it is y shaped, and this structure, the heavy chain is bind with the light chain by disulfide bonds and other non-covalent bonds, and it form the heterodimer structure. Okay, because it is having two type of chains like light chain and heavy chain, so that's why it is forming the heterodimer. Okay, then we will see like what is the anatomy of light and heavy chains. Okay, so firstly the light chain, it consist 100 to 110 amino acid are located at the end terminus, and the amino acid sequence varies among the antibodies. Okay, so the light chain end terminal region is varies among the antibodies. Okay, and this region of light chain is known as the variable region. Okay, so two type of constant region sequence are also found, that is called as the lambda and kappa constant region. Okay, in a particular antibody either two lambda or two kappa chain are present, but never it be like not one lambda and one kappa. Okay, if it is means in the light chain there is two region, one is constant, one is variable. Okay, so the constant region is can be two types. Okay, either it can be kappa or it can be lambda. Okay, so but it never be like one chain is consisting the kappa region and another is consisting the lambda region. Okay, so it should not be. Okay, so it is, if one immunoglobulin is there, so both the light chain is having either kappa or either lambda constant region. Okay, like that you are having in the antibody having one H chain. Okay, so in H chain about 110 amino acid are located and the N terminus region which shows greater variation among antibodies. Okay, this region is known as the variable region. Okay, so like light chain, heavy chain is also consisting the variable region. Okay, remaining amino acid sequence of heavy chain is somewhat constant, but reveal five different type of constant heavy chain region. Okay, that is mu, alpha, delta, eta and gamma. Okay, so based on the constant region there are five different classes of antibodies is present: IgG, IgA, IgD, IgE and IgG. Okay, IgM, sorry. Okay, then the length of the constant region of heavy chain is 330 amino acid for alpha, gamma and delta, and 440 amino acid for mu and eta. Okay, that is IgM, and Ig E is longer than these three different type of antibodies because it is having extra constant region. Okay, so yeah, this is the structure of antibodies. Here you can see that this is the constant, this is the heavy chain. Okay, this dark blue color, and this is the light chain. Okay, each chain is consisting one variable regions. Okay, here variable light, variable heavy, variable light, variable heavy. Okay, and then constant light, constant heavy, constant heavy, constant light. Okay, but in case of heavy chain there is three constant region is present, normal condition, but in case of M and E, there is four constant region is there. That's why the size is longer than the other antibodies. Okay, and this is made up of four polypeptide chain. Okay, and each is having two identical light and two heavy chains. All four polypeptide subunits are held together by disulfide bond and non-covalent bonds. Okay, and these two heavy and light chain is having it is categorized into three different parts. First one is the fab region. Okay, it is the antigen binding region, and it is present on the N terminal region, and effector function by the C terminal region of the antibody. Okay, and in antibody molecule there is two fab region are found and they bind antigen. Okay, so this region is called as the fab region. Okay, this is antigen binding site, this side and this side. Okay, these two are the fab regions because here only antigen binding happening. Okay, and the second thing is in the fab region there is the hyper variable region. Okay, on the light chain and heavy chain, which form the antigen binding site. Okay, so these two regions. Okay, these regions and this region, this is called the hypervariable region because it changes according to the binding. Okay, so that's why it is called as the hypervariable region. Then antigen binding site is complementary to the epitope of antigen, so it is called as the complementary determining regions. Okay, so this site is also called as the paratope because it is complementary to the epitope of antigen. Okay, so that's why it is called as complementary determining region, as well as the what I can say, it is the paratope. Okay, then you are having the FC region. Okay, this region is called as the FC region here. Okay, FC region. So what is FC region? FC region of immunoglobulin allow for interaction of immune complex with other phagocytic cell and complements. Okay, and means from the through this side the antibody will bind to any kind of cell surface and and initiate the immune complex. Okay, and take part in various biological functions that are determined by amino acid sequence of each domain of constant region. Okay, so which type of function it will do, it's depend on the amino acid sequence of each domain of constant region. Okay, constant region is having the means signal, means which kind of immune system it will participate. Okay, then it is having the hinge region. So the in case of IgG, IgD and IgA, heavy chain contain an extended poly peptide sequence between the CH1 and CH2. Okay, here. Okay, here CH1 and CH2 domain that has no homology with the other domain, this region is known as the hinge region. Okay, so this hinge region, what it generally do is it is rich with the proline residue. Okay, proline and cysteine residue, and it is flexible, so it provide the flexibility to bind the antibody upon the antigen epitope. Okay, so it in case of only IgG, DA is having this hinge region. Okay, IgM and IgE is not having this hinge region. Okay, so that you have to remember, hinge region is only found in IgG, IgD and IgA. Okay, so this is the structure of the antibody, where two chain is there, light chain, heavy chain. Okay, it is the heterodimer, and all this chain is having one means each chain is having one variable region, and light chain is having only one constant region, whereas this heavy chain is having three constant region in general cases. But in case of IgM and IgE, this is having four constant region. Okay, and it is having hinge region which provide the flexibility to bind the antibody on the antigen. Okay, then you are having different type of antibodies. So basically based on their constant region, there are five classes of constant heavy region. Okay, sorry, heavy chain. Five classes of antibodies is there. First one is the IgM. IgM is the first antibody produced in response to a microbial attack by B cell. Okay, so here it is the pentameric in structure. Okay, that you have to remember, it is the pentameric in structure. It is the largest antibody antibody, and it is found in pentameric form. It circulate in the body and lymph node, which consists 6% of the total antibody content in the serum. Okay, it is involved in agglutination and opsonization. It has a large number of antigenic site on its surface and therefore facilitate efficient activation of immune system, and it is found on the surface of mature B cell together with the IgD. Okay, so it is with the IgD, it is also found in the B cell cell surface. Okay, and it is first antibody to produce against any infection. Okay, then you are having the IgG, most abundant isotype in the plasma, and comprises 80% of the total antibody content in the serum. It detoxifies substances that are harmful and recognize the antigen antibody complexes. Okay, it is transferred to the placenta through the fetus and protect the infant until it births. Okay, so it is the only antibody which can cross the placenta. Okay, then it is divided into 4 subclasses: Ig1, Ig2, Ig3, 4. Okay, among these only Ig3 and 4 possesses the ability to cross the placenta. Okay, the heavy chain of IgG have 2 antigen binding site and are of subclasses gamma. It facilitates the process of phagocytosis and provide immunity to the developing fetus. It neutralize the toxin and pathogen and offers a protection to the body. And there is 4 classes of subclasses of IgG. Okay, and this is consisting of normal immunoglobulin structure. Okay, and IgA, third antibody is IgA, so usually found in the liquid such as breast milk, serum, saliva, fluid of intestine. IgA in breast milk protect an infant gastrointestinal tract from microbial activity. Okay, it constitutes 13% of the total antibody content in the serum, and it divided into 2 subclasses, Ig1 and Ig2. Among these, Ig1 is highly found in the secretion and is also called the secretory immunoglobulin. Okay, IgA is called as the secretory immunoglobulin. It can exist monomeric as well as dimeric form. Monomeric form is present in the serum, whereas this dimeric form is present in the secretory form. Okay, this IgA present in the serum predominantly in monomeric form. It may provide the neonate with a major source of protection against pathogen during the first week of after birth. Okay, so here it is the dimer structure of the IgA, which present in the secretory form, whereas in the serum form it is present as the monomeric form. Okay, then you are having the IgD. IgD is involved in the production of antibody by B cell. It is present as a monomer and weigh around 1,80,000 Dalton. It comprises less than 1% of the total antibody content in the serum. It acts as a receptor on B cell surface and participate in B cell activation and differentiation. Okay, so the biological function is not known for the IgD, whereas it is present as a receptor molecule on the B cell surface, or you can say that it is the B cell receptor. Then you are having the IgE. IgE is present in the in the least amount, around 0.02% of the antibody content in the serum. Okay, these are present in the lining of the respiratory and intestinal tract and respond to the allergic reaction. That you have to remember, it is responsible for the allergic reaction, and this is found as a monomer in the body and weight around it is 2 lakh Dalton. Ig bind to the Fc receptor on the membrane of basophil and mast cell to provide immunity against allergic reaction. Okay, so IgE mediated degranulation is necessary for the anti-parasitic defense. Okay, so here you can see that this is the IgE. Okay, which is contain here the 4 constant chain is there, you can see. Okay, so this is the IgE. So these are the different classes of antibodies, their properties, and this is the structure. So Ig is basically found in the secretory form. D is present on the B cell receptor, activate basophil and mast cell, and Ig protect against parasitic worm and responsible for allergic reaction. Okay, and IgG is secreted by plasma cell in the blood, able to cross the placenta into the fetus. Okay, and IgM may be attached to the surface of B cell and secreted into the blood, responsible for early stages of the immunity. Okay, so it is pentameric in nature, and this is the dimeric in nature. Remaining will be monomeric in nature. So in this question it has asked, like, based on the heavy chain, which one of the following antibodies has the multiple subtypes? Okay, IgM, IgD, IgE and IgG. What will be the correct answer? We will see, right? So, in the previous slides, we have seen that this IgG and IgA is having the subtypes. Okay, in case of the IgG, there is four sub classes. One is IgG1, IgG2, IgG3, and IgG4. Okay, and among these four subclasses, only IgG3 and IgG4 possesses the ability to cross the placenta. Okay, so IgG3 and IgG4 is main subclasses which can cross the placenta, or remaining two IgG can't cross the placenta. Okay, and this IgG is having four subclasses. Okay, and like that IgA is also having two subclasses, IgA1 and IgA2. Okay, and among these two subclasses, IgA1 is highly found in the secretion and is also called as the secretory immunoglobulin. Okay, so whatever secretory antibody is there, that is mainly the IgA1. Okay, not IgA2. Okay, it is the IgA1 which is having the more concentration in the secretory antibody. Okay, and it is called as the secretory immunoglobulin. Okay, so in the given option, it is only mentioned the IgG. Okay, so here the IgG will be the correct answer. Okay, because IgM is not having any kind of subclasses, and this IgD is only the biological function of IgD is not known. Okay, and only the IgD work as a B cell receptor. Okay, and this IgE is basically for the parasitic infection or allergic reaction. Okay, so this IgG and IgA is having the subtypes. Okay, so in the option only IgG is given, so that's why this is the correct answer. Okay.